Let's talk about plasma membrane. This is the previous version of the ultrastructure of plasma membrane. There is a new model commonly known as the fluid mosaic model, which I will discuss in my next video. Presently I am discussing the preliminary model of the plasma membrane. So plasma membrane is thin, electron microscopic. Electron microscopic means it can only be seen under electron microscope. It is transparent, regenerative. It can self-regenerate itself, and semi-permeable in nature. Semi-permeable, it means it selectively permits specific molecules in and out of the cell to maintain cell homeostasis. So plasma membrane is also referred as plasma lemma. This is important. And is found in both prokaryotes and eukaryotes, that is old cells like bacteria and human cells as an example of eukaryotes, animal cells. Now about biomembranes. So both in eukaryotic cells, the cell organelles which are present inside the cell are all membrane bounded, and this plasma membrane, which is the outer covering of the cell, and the membranes which covers the cell organelles have same ultrastructure and are collectively called as biomembranes. What are the functions? It regulates the exchange of the materials inside the cell, in and out of the cell, to maintain cell homeostasis, that is a regulation in the cell, and thus helps in the synthesis of different macromolecules. Molecules are sometimes called as moieties. Now the biochemical composition of the plasma membrane. Human RBC was first chosen by some Dutch scientists to explore the structure of the plasma membrane, and it is taken human RBC. Why? Because human RBC lack nucleus and cytoplasmic organelles, so it is very easy to explore the ultrastructure of the plasma membrane taking human RBC. Let's discuss the structure. You see, these are the proteins, and this is also proteins. Here what you see, are lipid moieties. That is an example, common example of lipid moiety is lecithin. This lecithin or lecithin is found in between this protein moieties or the molecules, and over here are the cholesterol molecules. So an enlarged version of this is given here. This is the protein, this is the lecithin or the lipid moiety. Alongside we have the cholesterol, and you see these lecithin moieties are facing other lecithin moieties opposite to each other. Okay, so this is the basic concept. New version or the new concept of the ultrastructure of the plasma membrane is described in the fluid mosaic model, commonly named as fluid mosaic model, which I will discuss in my next video. This is just the basic demonstration of the plasma membrane. So lipids, as I described earlier also, this structure, lipids present with cholesterol moieties. Let's take different colors. You see, these two lipid molecules are facing each other. So does the cholesterol moiety. Let's take this color. So does the cholesterol moiety. Now, what are lipids? So, cell membrane contains lipids. These purple colors are the lipids, and bilayer arrangement of the lipid molecules. This is not liquid, this is actually lipid, sorry. So these are bilayered in nature. And now an important term, amphiphatic nature. What is amphiphatic? Polar or the hydrophilic head of glycerol of the phospholipid molecule of two layers are directed in opposite direction and interact with aqueous environment on both sides, and non-polar or hydrophobic tails of two layers face each other. So let's use a black color. So these are the polar heads of the lipid molecule. These are polar or hydrophilic. Hydrophilic means they can face the water molecule. So this polar or the hydrophilic head of glycerol of these phospholipid molecules are directed in opposite directions and interact with the aqueous environment. This is the aqueous environment, so this can interact. So let's consider this as the cell. So plasma membrane is obviously outside to cell, and this is the cell nucleus. So this plasma membrane is up to this. Okay. And this is bilayered arrangement. This polar or the hydrophilic head are placed in opposite direction because outside of the cell there can be, the medium is aqueous, and also in the cytoplasm, this is the cytoplasm, right? So this medium is also aqueous. It has to be aqueous in nature. Cytoplasm is aqueous only. So if these heads are hydrophobic, then they will not stay in this bilayer arrangement. It is the hydrophilic portion of this lipid moieties or the molecules which helps to retain this bilayer structure of the plasma membrane. And likewise this non-polar hydrophobic tails are facing each other. These hydrophobic tails are facing each other. So this is the assemblage or the ultrastructure of the plasma membrane, preliminary described by some Dutch scientists. Now phospholipids. Phospholipids accounts 55 to 75% of the total lipids in the plasma membrane. This is very important. And common types I have mentioned is the lecithin, or whatever can be pronounced lecithin, cephalin, phosphatidyl ionositol, and phosphatidic acid. So these are some of the common types of phospholipids, but lecithin is the abundant. So phospholipids forms the structural framework of the plasma membrane, as I mentioned or described in the previous slide, and allow movement of fat soluble molecules. So fat soluble molecules can trespass this membrane because of these phospholipids molecules. Cholesterol molecules, as I have mentioned, if this is the lipid molecule, phospholipid, alongside we have the cholesterol. This is the cholesterol moiety, and it accounts 25 to 30% of the total lipids, and it lies between the phospholipid molecules. So if this is cholesterol, this is phospholipid, so again there will be one phospholipid. So it is lying between, this cholesterol is lying between the two phospholipid molecules, and thus stabilizes the plasma membrane or the cell membrane. So let's discuss about proteins. As I have already mentioned, let's draw once again with this color. These are the protein moieties or the molecules. And here we find the phospholipid, okay. This is again protein moieties, because plasma membrane is bilayer in nature. It has two layers, and this is also a phospholipid, and between these phospholipids we have the cholesterol molecules, right? This is the basic configuration. So proteins, these red ones, proteins, accounts for 58 to 59%, and the ratio of proteins to lipid varies from cell to cell. Membrane bound proteins are of two types on the basis of their association with the phospholipid molecule. It can be peripheral or the extrinsic proteins, and intrinsic or integral proteins. This is very important. Proteins found in the plasma membrane are basically of two types: peripheral, as the name suggests, peripheral means found in the periphery of the plasma membrane, or extrinsic proteins, and intrinsic means found inside, very near to the cytoplasm, or integral proteins. Now peripheral proteins, its location is outside the phospholipid. Peripheral proteins location outside the phospholipid bilayer can be easily separated with salt solution. This is important. So actually this is a procedure to lyse a cell. Lyse means to break apart the cell. So peripheral proteins, as the name suggests, is found in the periphery of the cell, that is outside the phospholipid bilayer. So if you want to break the cell and extract the DNA inside the cell, so peripheral proteins can be easily separated with salt solution. You just apply salt, the protein will get denatured easily, or using some chelators. So peripheral proteins accounts 30% of the proteins. Some common examples which is very important: spectrin found in the RBC, acetylcholinesterase present in the neuron, and ATPase present in mitochondria. So spectrin, acetylcholinesterase and ATPase, these are very important peripheral proteins, and you will often find these names in biology, microbiology and biochemistry, biotechnology as well. Spectrin, acetylcholinesterase and ATPase. These are peripheral proteins. Some examples. Next, intrinsic proteins. These are embedded in phospholipid bilayer. Can be separated by detergent solutions. After you apply salt, the extrinsic proteins will go away. Next, you have to apply detergent solution to expel these extrinsic proteins. So it accounts for 70% of the membrane proteins. Some examples of intrinsic proteins are cytochrome oxidase and rhodopsin. These have their own structures which is not so important as of now. Now, structural proteins. This forms the backbone of the membrane. And what does it do? It gives mechanical stability to the cell and also elasticity. Examples: enzymatic proteins of plasma membrane. It controls cellular activity. Carrier proteins helps in active transport, as the name suggests, carrier, so it will help in active transport. And enzymatic protein helps in cellular activity, and hormone receptor protein, that is the action of hormones. So structural proteins are important. Now in the plasma membrane we also find some carbohydrates moieties. Some examples are hexoses, fucose, sialic acid, hexosamine. And these carbohydrates are associated with proteins to form glycoproteins and glycolipids. Helps in cellular recognition, blood grouping and immune response. So carbohydrates are actually helping the cells for recognition, blood grouping and immune response. So I think this is the overview of the plasma membrane. Actually, when I will discuss the fluid mosaic model, that would be the appropriate one, and this is just the preliminary ideas given by some Dutch scientists. So more advanced level is the fluid mosaic model of the plasma membrane, which I will discuss in the next video.